Hi, Michelle Glass here. Welcome to another one of our videos for Chapter 19, Lecture 19. We are finishing up here our discussion of those formed elements that we see in the whole blood. So remember, when we are talking about the whole blood, a little less than half, about 45%, is composed of formed elements. And there are, remember, three types of formed elements. We have already talked about our red blood cells or erythrocytes. We already have videos talking about white blood cells or leukocytes, and our presentation here is focusing in on those platelets, which we can also call by this other name, thrombocytes. Now, platelets, like red blood cells, are not fully intact cells. What we see with our platelets is that they are actually cell fragments. So there is a cell type called a megakaryocyte, mega for big, karyocyte for nucleus. So these are big nucleated cells. And what we see is that they break off into these cytoplasmic fragments. And because they're fragments, they, again, like red blood cells, they don't have a complete complement of organelles, so they can't do any kind of repairs or anything like that. And so what we see is that they will be in circulation for about 9 to 12 days, and then you have some phagocytes coming along and gobble up those platelets as they're starting to go past, they're starting to expire, we could say. Platelets are going to be important in formation of our blood clots, so they will be releasing some chemicals to help that trigger that process. Blood clotting, remember, is that classic example of a positive feedback mechanism, so we will have a video dedicated just to that topic. We also will see our platelets involved in making temporary patches. That patch we will be calling a platelet plug. So as part of this process of forming the blood clot, you're going to put a little temporary plug in there first while you're getting that whole clot to be formed. And then we'll see also that platelets are important in helping to contract and squeeze the walls of those vessels that are broken closer to each other to decrease the size of the break. So they're kind of like our first responders to our damage of a blood vessel. So they're going to help to make that break smaller. They're going to give us a temporary plug, and they're going to release some important chemicals to initiate that blood clot formation. And so stay tuned for our last video looking at that process of making a blood clot, what we can call collectively hemostasis, where that prefix heme means blood and stasis means like standing, same as homeostasis, but in this case, hemostasis really just means stop blood loss, stop bleeding. Okay, so stay tuned.